Kelyah Spurgeon. So Kelyah is currently a master's student at North Carolina A&T State University, and she was awarded an NC Space Grant graduate research fellowship in the 2021-2022 school year. She conducted research assessing how microgravity affects the evolution and behavior of Streptococcus mutans biofilms. So the floor is yours, take it away.
Good afternoon, everyone. My name is Kelyah Spurgeon. I'm a second-year master's student at North Carolina Agricultural and Technical State University, working in the Thomas Lab. My research aims to assess the impact of simulated microgravity on the adaptation of Streptococcus mutans biofilms. I'm sure a lot of you have heard by now about the ambitious manned spaceflight missions planned for the future. Unfortunately, there's still a lot we don't know about how spaceflight affects our bodies. We know that humans need water, air, and food to survive in space, but we also know that there are morphological and biological changes that our bodies undergo in space, such as decreased bone marrow absorption and decreased salivary lysozyme production. However, we still don't know why astronauts display an increased rate of dental plaque buildup leading to dental disease. In 2012, it was predicted that dental caries would be among the top 5 medical conditions that would negatively impact future space missions, and not much is known on why this risk exists. Here on Earth, Streptococcus mutans is known as the etiologic agent of dental caries. Formation of biofilms facilitated by S. mutans can lead to severe dental diseases like cavities, gingivitis, and periodontitis. S. mutans resides in the mouth as a commensal microbe for humans, but its virulent state on the right side of the black and white photo may be responsible for the high rate of dental caries we see in space. Current studies have looked at oral species in simulated microgravity, including S. mutans. However, these studies are limited in that most of them are short-term studies and cannot be applied to extended spaceflight missions, or they assess long-term adaptation, but only represent a single SMG population with no normal gravity controls. The way that we simulate microgravity in our lab is a method commonly found in the literature. We use high aspect ratio vessels, or HARVs, where the bacteria and the surrounding media can experience weightlessness. The Earth's gravitational force in the device is reduced from 9.8 to 0.5 meters per second squared. HARV motion is depicted here in the loop video. We found that S. mutans biofilms are typically observed on hydroxyapatite, a solid material responsible for surface enamel repair in humans. We combined these studies to form a new protocol that can be used to study S. mutans biofilms in SMG. So how exactly did we accomplish this? Initially, we had to decide on what type of hydroxyapatite material to use for the study. Our options were either hydroxyapatite beads or hydroxyapatite discs. We wanted an HA medium that could provide accurate and reliable results while properly supporting the biofilms over the course of 100 days. We found that HA beads could be used to gather data, but beyond 21 days, the results became inconsistent. The adhesion data from the beads demonstrated a greater number of live cells on the HA beads and normal gravity control seen in blue over the microgravity treatment in pink after 21 days. We then switched to HA disc and we found that removing the biofilms from them was much more efficient and the results were consistent for use over 100 days. The variation in adherence data seen on the right from the disc is similar to what we would see in publications. It's consistent with published data and our own planktonic data currently under review. So we decided that HA discs were the best way to move forward. And so we moved to the next step, which was adaptation. We knew that we wanted to include both normal gravity and microgravity treatments, but we weren't sure about subculturing the media with the HA disc attached inside. We tested methods to do this, and we found that HA disc adhered to the inside of the HARVs, and they would support S. mutans biofilms through the course of 100 days, as pictured here. Our final step was to develop assays for both phenotypic and genotypic changes associated with S. mutans biofilms adaptation in SMG. We created a process in which the biofilms could be removed from the HA disc and analyzed for various assays, including acid stress tolerance, oxidative stress tolerance, and adhesion. The biofilms removed from the disc could also be used to perform genomic extractions. Here's an example of acid tolerance data that we gathered from the HA disc using the protocol I just described. In summary, over the course of this study, we've been able to successfully develop a new method to grow biofilms in SMG. Our protocol can be used to assess long-term adaptation, phenotypic changes, and perform genomic extractions to find data on biofilms simulated in microgravity. Thank you.
I do have a question for you, Kelyah. Are you guys planning to further develop this and maybe do a parabolic flight or a drop tower test or some type of longer or other microgravity test?
Actually, Tyler, I'm not familiar with either of those terms. Right now, we're just looking for multi-species biofilms as the next step, just simulating the same microgravity environment with the HARVs with more species.
Okay, yeah, so little plug here, but NASA Glenn actually is home to two of the drop towers that are owned by NASA facilities. So we use that a lot, at least in my group, for testing combustion or fluidic systems, and it does give you a kind of a higher degree of microgravity than you can sometimes get in the lab. But I have seen, similar setups to the one you had there and your diagram, mainly for the biological side, especially biofilms and things like that, but even for plants and related work like that, those are fairly common. And there's one thing, I'm not sure if you've come across this or not, but one of our little fluidic tricks for getting things to work in microgravity is what we use. It's called a Hellishaw cell. It's almost like a petri dish turned on its side. I was wondering if you came across that term at all.
No, we haven't, but I know
Okay.
that we've used other forms of simulating microgravity besides the HARVs.
Mhm.
That's
Yeah,
pretty cool.
it's very similar to your concept, but I was just curious. Great. There is one question I see in there. So in what ways, if any, has conducting your research validated, refined, or even changed your career instincts?
So I was always interested in microbiology, but this whole simulation of microgravity got me interested into biofilm dynamics overall. So now I'm looking forward to studying just biofilm communities on an ecological side.
Gotcha. Great. Well, I think we're going to have to move on at this time, but thank you again. It was a great talk.